You can start, Liad.
Thank you. Welcome everyone to the 2025 Coller-Dolittle Challenge for Interspecies Two-Way Communication. My name is Liad Mudrik and I'm a researcher at the School of Psychological Science and Segol School of Neuroscience at Tel Aviv University, and today I will be your host in this event. Inspired by the Turing test, the Coller-Dolittle Challenge calls on the scientific community to develop algorithms for decoding non-human communication and conducting two-way interspecies communication, initiated by the Jeremy Coller Foundation in partnership with Tel Aviv University. The $100,000 prize will be awarded every year in the next 5 years to a team that made most progress in obtaining the following criteria. First, using a non-invasive approach to communicate with non-human animals or decipher non-human communication. Second, demonstrating communication in more than one context, like alarm, mating, foraging, using the organism's endogenous communication signals, preferably in an interactive and autonomous way, and demonstrating a measurable response of the organism to the signals broadcasted to it. The 4 finalists were selected following a 2-step procedure. First, the submitted proposals were evaluated by external scientists and with relevant expertise. And next, the external reviews were reviewed by the scientific committee, and the 4 with the best evaluations were selected. Today, we'll hear from our 4 finalists who will be showcasing their groundbreaking research on interspecies communication. Their work pushes the boundaries of science, deepening our understanding of animal language and cognition. We look forward to an engaging and insightful afternoon as we explore these exciting proposals. Thank you for joining us. And now we will get to know our judging panel. Hear from Jeremy Coller, co-creator and patron of this initiative. Listen to the 4 finalists showcasing their research and enjoy a lecture by our special guest, Dr. Irene Pepperberg, before the judges announce the winning team or teams for the 2025 installment of the Coller-Dolittle Challenge. So with us are 8 esteemed judges. Please turn on your cameras so we could see you. Yossi Yovel, Professor of Zoology from Tel Aviv University, who is also the co-creator of this initiative and spokesperson for the judging panel. Elodie Briffer, an ecologist from the University of Copenhagen. Amir Teicher, Professor of History of Science from Tel Aviv University. Jonathan Birch, Director of the Jeremy Coller Center for Animal Sentience in London School of Economics. Miriam Knorschild, biologist from Freie Universität Berlin. Inbal Arnon, Professor of Psychology from the Hebrew University of Jerusalem. Oded Rechavi, geneticist from Tel Aviv University. And Raja Jairis, AI researcher from Tel Aviv University. It's a pleasure to see you all. Again, turn on your camera so we could see you. Many thanks to all of you for agreeing to serve as judges, and please feel free to turn off your cameras now, because we would like to hear now from Jeremy Coller. I would like to introduce Mr. Coller, who is going to talk to us about what we know, what we don't know, and how we can minimize the gap between these 2 things.
Hi, Liad. Thank you. Hello, Liad. Thank you. Thank you, everyone, for joining us today for the first time of what I'm sure will be many significant steps towards a very exciting future. We've heard a lot from successful business leaders who think the future lies in stars, exploring space, setting their sights on Mars even. And that's great, really looking forward to seeing it. But in pursuing those lofty goals, we shouldn't lose sight of the fact there's still so much we don't understand here on Earth. And that's what's inspired me to create this challenge, asking humanity's finest scientific minds to take the latest technology and insights and use them to unlock the mysteries of interspecies communication. We actually don't know how many animals we share this planet with. Some estimates say up to 8.7 million different species. I don't know why it's not just 9, but million, but 8.7 million species, and as many as 20 quintillion individual creatures, from the tiniest insect to the mightiest whale. And yet we as humanity have had no interest in what any of them are saying to one another, or even us. We're an extraordinarily speciesist species. We've arrogantly convinced ourselves that we are the only living things worth listening to. But if you don't listen, you'll never hear. And if you don't look, you'll never see. I'm so absolutely delighted to say that today's finalists have listened and they have looked and they have already shown the world's things nobody knew before. Of course, we're not yet at the point where we can parley with pachyderms or discuss Eastern art and dramas with intellectual llamas. And we're a long way from my personal goal of giving a voice to the animals trapped in our horrific and unsustainable food system. But the work we're celebrating today, fascinating and vital in its own right, also moves closer to a time when we can listen in on the previously unheard conversations going on all around us, and maybe even take part in them ourselves. After all, for well over 1,000 years, we assumed that hieroglyphics would never be deciphered. Many people said they couldn't be translated at all because they just represented no kind of coherent language. They were just symbols. Does that sound familiar? And then, of course, in 1799, Pierre-François Bouchard stumbled upon the Rosetta Stone, and with that key, it took barely 2 decades to unscramble texts that had not been read for millennia. In 2025, technology such as AI offers us any number of Rosetta Stones, the ability to open countless doors that were previously locked. In 2000, we could combine that with the kind of innovative, boundary-pushing work we are celebrating today, and I think we can all agree that we're getting closer to something very, very special indeed. I feel humbled by all the people who have helped us reach this point. I'd like to thank many, many people, but first of all, to Yossi and all the judges for everything they've done. It's amazing. Gaia and Gavin and Jorge and Alex and Gary, without you, this whole event wouldn't have been possible. So thank you for in the background making all of this possible, and congratulations to all of the finalists. I don't think we're going to claim victory today, but as long as you keep working on this, the whole planet wins. Liad, back to you and the 4 finalists. Thank you.
Thank you so much, Jeremy. Thank you for these inspiring words. I think it's now indeed time to hear from our finalists. So all presenters, I just want to make sure the audience knows, all presenters have some audio or video materials to share. So please ensure the volume is on, your device is up. According to your internet connection, you might experience some delays or unclear sounds, but worry not, because this event is being recorded and you will be able to watch it on demand soon after the live event. Also, if you have any questions for our speakers, please feel free to type those in the Q&A window, and I will present these questions to the speakers after their talk. Our first presenter is Leila Sayag, Senior Research Specialist from Woods Hole Oceanographic Institution. Leila will present her team's research, First Evidence of Widespread Sharing of Stereotyped Non-Signature Whistles in Wild Dolphins, co-authored by Franz Jensen, Katie McHugh, Marco Cazzoli, Laurine Lemercier, Peter Tayak, Randall Wells, and Vincent Janek. Leila, the stage is yours.
Thank you so much. I'd like to start by acknowledging my co-authors, without whom this work would not be possible. And we're really honored to be finalists for the Coller-Dolittle Prize. And I'm really excited to be here today to tell you about our research. So can we talk to dolphins? Dolphins are ideal. Oh, I'm not ready for the next one yet, but that's okay. Dolphins are ideal candidates to meet the goals of the Coller-Dolittle Challenge. There are numerous parallels between their communication system and ours. Next slide. So these evolutionary parallels to our language include the fact that dolphins are flexible, lifelong vocal learners. They produce learned name-like signature whistles, which are copied by other dolphins to initiate contact, similarly to how we use our names. Female dolphins use motherese or baby talk when communicating with calves in a way similar to humans, and dolphin whistles have the potential to function referentially. Next slide. So these findings and others, back to the previous one. These findings and others have come from over 40 years of research by our team, and some of our publications are listed here. Next slide. So these are 12 examples of signature whistles or dolphin names. Each is from a different dolphin labeled with their identification numbers, and you can see some of their distinctive contour shapes. And these whistles are stable over time, as shown by the images on the right, which are examples of the signature whistle highlighted in red, recorded over a period of 10 years. And these distinctive names are developed with vocal production learning, a feature that dolphins share with us. Next slide. Most of what we've learned about signature whistles has come through a collaboration with the Sarasota Dolphin Research Program, which is the longest running study of a population of wild cetaceans in the world. This resident community of approximately 170 wild dolphins in the waters in and around Sarasota Bay, Florida, USA, spans up to 6 generations, and almost all individuals are of known age, sex, and relatedness. This project uses unique research methods that have enabled us to get around one of the most challenging aspects of studying dolphin communication, which is identifying which dolphin is making a sound. Next slide. We use non-invasive recording methods during brief catch and release health assessments, and these include recording known individual dolphins using suction cup hydrophones while they're briefly being held, and with suction cup attached digital recording tags that the animals wear for up to 24 hours after being released. And these non-invasive recording methods enable us to identify which dolphin is making a call, resulting in a highly unique data set. Next slide. Yeah, there we go. Signature whistles produced by dolphins during health assessments are typical of those that they produce when free swimming. And this image shows whistles from 4 different dolphins recorded in these 2 contexts. These examples also highlight some important features of dolphin signature whistles. Many contain repetitive elements called loops, which may or may not be connected to each other, as shown in the first 3 rows. And sometimes they're distinguished from other whistles by very subtle features, such as those indicated by arrows in these images. And so, for example, looking at the second loop of Dolphin B's whistle, you can see 2 subtle inflections that she preserves whenever she produces her whistle. Next slide. And so over the past 4 decades, we have built the Sarasota Dolphin Whistle Database. And shown here is one signature whistle or name from each of 269 individual dolphins. And this large library of high-quality recordings is unlike any other data set in existence for a population of wild cetaceans. Next slide. In Sarasota, about half of the whistles dolphins produce when free swimming are signature whistles. What about the other half? Next slide. These are called non-signature whistles, and non-signature whistles are very difficult to study if the signature whistles of a population are not known and have therefore received little attention. But since we know the signature whistles of most dolphins in the Sarasota community, we have a unique research opportunity. Next slide. We have now identified more than 20 repeated shared non-signature whistle types. And this image shows 8 types, with each column being a different whistle type, and the example in each row produced by a different dolphin. Next slide. Some non-signature whistle types are unusual, containing features that are rarely seen in signature whistles. 2 of these are shown here. When I first saw whistle type A, shown on the left on a tag recording, I didn't actually even think they were dolphin whistles at first. These steep or short up and down sweeps were so different from the signature whistles I was used to studying. But now that we started looking for this whistle type, we've seen it produced by 25 individuals so far and still counting. And these 4 examples shown here were produced by 4 different dolphins, as shown by their identification numbers on the spectrograms. Whistle type B on the right is characterized by a variable beginning portion followed by a characteristic flat or constant frequency component, which is very unusual in Sarasota dolphin whistles, signature whistles. I was really surprised when I first saw this whistle type produced by multiple animals in response to non-signature whistle playbacks. And since then, we found it produced by at least 35 different individuals and still counting. And again, the examples shown here are produced by 4 different dolphins with the ID numbers shown on the spectrograms. Next slide. So what are the functions of non-signature whistles? We're studying this with playback experiments. We play back natural whistles to dolphins underwater and film their reactions with drones. And as you'll see in the video I'm about to show, we observe a variety of behavioral responses to whistle playbacks, which give us insights into how these whistles function. So next slide. So in this approach response, you can see the speaker icon at the bottom, hopefully, and hear the whistle. And you can see that the dolphins are turning toward and approaching the playback stimulus. In this avoidance response, the dolphin turns away from the stimulus and swims away from the stimulus. In this interest response, the dolphin turns toward the stimulus and stays in place, investigating it. And in this ambiguous response, the dolphins initially turn away from the stimulus and then turn toward it. Okay, next slide. So the varied responses that we observed reflect a complex communication system. Just as with humans, we wouldn't expect a one-size-fits-all response to any sound type, even our own name. Our response would depend on many things, including our recent interactions with others. So this means we need a lot of data to be able to correctly interpret responses, but it also makes them much more interesting. But some trends have emerged from the data that we have so far that provide insights into how some of these whistles function. Next slide. Our controls are self-signature whistle playback, so the signature whistle of the dolphin we're playing to. And these mostly elicited approach, affirming the function of signature whistles as a way of initiating contact. Next slide. The up, down, up type A whistles mostly elicited avoidance responses, suggesting an alarm type function. Next slide. And the type B whistles with the flat component elicited varied responses, supporting our hypothesized function of this whistle as a query produced when something unexpected is heard. Next slide. Our work meets the criteria of the Coller-Dolittle challenge because it uses a non-invasive approach to decipher dolphin communication, demonstrates communication in more than one context using the dolphin's own signals, and demonstrates a measurable response to whistle playbacks. Next slide. The Coller-Dolittle Prize would enable us to continue our AI and machine learning analyses, which are focused on whistle classification, enabling us to build our database of the dolphin vocabulary. And we would also expand our tests of whistle function. Next slide. So in closing, dolphins are ideal candidates for the Coller-Dolittle challenge. There are many parallels with our language, include flexible, lifelong vocal learning, use of motherese or baby talk, learn distinctive name-like signature whistles that are copied by other dolphins to initiate contact, and potential word-like non-signature whistles. These parallels could enable us to build bridges between our communication systems, which we're excited to continue exploring. Next slide. This work has involved many people and funders over the years, which are too many to name, but I do want to give a shout out to our current funder, the Avatar Alliance Foundation.
Thank you so much, Leila. This has been fascinating, and we have a few minutes for some questions. First, I want to ask you, you presented this question at the end, can we talk to dolphins? Do they ever talk back? So do they reply to the sounds that you present to them?
Yes, I'm glad you asked that. We have seen not many vocal responses actually to these free swimming playbacks. We've seen a couple really interesting examples, though. In one case where we played back kind of a very unexpected scenario to a pair of males that were together, we played back one of their own whistles, and they made that non-signature whistle B in response, which we, was sort of interpreted as kind of a surprise, like what is that? And we have also seen other dolphins engaging with playbacks that we've done during health assessments where they've kind of called back and forth with the stimulus. But in a lot of our free swimming playbacks, we actually don't see responses. And we're not really surprised by that, since we're, like often playing kind of unexpected whistles, and dolphins may not want to sort of announce their presence until they've had a chance to fully investigate what's there.
Interesting. And do you see consistency either in the vocal responses or in the behavioral responses, both within individual or between individuals?
We don't have enough data to speak to that yet. So we are trying to get multiple playbacks with the same individual, and as well as with as many different individuals and contexts as possible, because we really think context is very important, like whether they're with other dolphins at the time, whether they're alone, what they're doing, also their history of interactions with other animals. So it's something that is quite nuanced, but is going so it's going to require a lot of data. But again, that's kind of what makes it very interesting.
Is why you're here, basically, to get that data. So maybe last question, what is the place of the AI in this project? So how elaborate is the use going to be in AI?
Yes, very important. We're actively working with computer scientists. We have been for the past several years on using our large database of known whistles from known dolphins to train AI algorithms to classify dolphin signature whistles. And this requires a lot of training data because the whistles have a lot of natural variability in them. But we have that with our amazing whistle database. And so once we're able to accurately use AI to classify signature whistles, the next step will be to then develop AI algorithms to classify non-signature whistles, and that'll enable us to process much larger quantities of data. For example, we have listening stations throughout the study area in Sarasota that are recording continuously. So if we can capture those whistles and classify them automatically, we'll have a really amazing data set.
Wonderful. We are more or less out of time, but there are many questions in the Q&A, so maybe I'll just pick one of them, and hopefully we'll get to answer the others maybe in another occasion. So the question from Ralph Peterson is, have you attempted playing back vocalizations from their own versus other pod? And if so, do you see any different behavioral responses?
So yes, that's a great question. And we have done a few playbacks of unfamiliar whistles recorded from other populations. And all I can say is that we've seen varied responses to that. So not very satisfying yet. It's, I think, similar to like the non-signature whistle playbacks, where it's probably going to depend a lot on context, how they might respond to an unfamiliar whistle. For example, males might be more interested in a potential newcomer in the population than females who might not be. So we have more work to do to look at that, but very interesting question.
Thank you so much for this interesting presentation and for the answers. And it's now time to go on to our second presenters. So thank you again. Next up are Daniela Valentin from the research group leader at the Max Planck Institute for Ornithology, and Jan Clemens, a group leader at the European Neuroscience Institute, Göttingen. And they will present their team's research, Can We Talk to Animals? Scientists Decode Nightingales' Complex Songs.
Thank you very much. We're excited to share our collaborative effort between a mathematician and a biologist to decode nightingale song. And if you show the next slide. Okay. So we're excited to be here, and okay. So nightingales are renowned songbirds for their complex singing behavior, and they actually have been cited in Shakespeare in Romeo and Juliet for their romantic singing. But they do not only sing for romance, but they also engage in fierce counter-singing duels between two males in order to defend their territories. And to showcase one nightingale singing here, this is actually a rare sight, I will play you this video where you will hear a nightingale singing a few complex songs. Next. So I hope you could appreciate this beautiful singing. And we thought that this nature's vocal specialist is actually ideal to tackle the Dr. Dolittle challenge, where there are three criteria to understand and to communicate with animals. So the first one is that you have to use their own vocalizations to interact with them. And here nightingales are ideal because they have a really large vocal repertoire of about 270 different songs each bird. The next challenge would be to communicate with them in different contexts. And here nightingales, as I alluded to before, they're singing to attract females, but also to defend their territories. And the third challenge would be, and this is probably the most complex one, is to really put meaning onto their song and what they are saying. And here we will use songs for long-range communication, aggression, and attraction. Next. So to dive a little bit deeper into how nightingales are singing, I will show you two nightingales counter singing with each other. And there's actually something special about their behavior here because they're performing song matching. And song matching, what that means is that one nightingale is really actively listening to what the other one is singing and then replicating or copying the exact same song. And this is the readout basically that we can use to understand whether the nightingale is communicating with us. So in the following, you will hear two nightingales singing. You will also see the spectrogram, which is the frequency representation of the sound. You saw this in the previous talk already. And then you will see hear the song unfolding. Next. Next. So I hope you can see and appreciate it, and also acoustically, that the last two songs were really the same songs. Next. So now having this behavior at hand, what we did first was to dive into the vocal repertoire, meaning we, using machine learning tools, we dissected all the syllables of the nightingales and were able to classify them in distinct clusters. Next. And with this syllable alphabet at hand, we were able to create a nightingale dictionary. It's of course not a nightingale dictionary in the classical sense because we cannot put sense or the meaning to these songs yet, but we at least can classify all the different songs. So having this at hand, we wanted to see how we can communicate with the nightingales, and we picked one of the syllables from their vocal repertoire that was in this case also a whistle, an unmodulated frequency sound that we can also produce. Next. Yeah. So what we did is, or what in this case John did, he went into the wild and he started whistling to a nightingale because we as humans can also whistle. So let's see what happened. Next. Next. So I hope you can see or also heard that the nightingale was responding with a whistle, and it was also responding with almost the same pitch. So we wondered, okay, how reliable is this behavior? And that's why John whistled again. Next. Next. And here you can see that the bird also replied with a whistle with the exact pitch, but it also added something on top, what is probably very difficult for John to whistle. But then, since this is a territorial defense behavior, we thought, do we have to leave or can we stay? So John whistled again. Next. One more next. And next. So here, as you can see, we had to leave the territory because this was a perfect pitch match by the nightingale. But these were just preliminary data. Next. And to really create an experiment from this, next, we actually went into the wild and next, we conducted controlled fieldwork experiments in the wild with nightingales during the night, because this is when they're singing. So here Sebastian and Giacomo went out and played many different whistle playbacks to the nightingales and saw how they responded. Next. And long story short, nightingales match the pitch, but also the duration of these whistles. And just to give you a short glimpse of the experiment, I will now play 3 examples of this beautiful interaction. Next. So the last one was probably a little bit more complex. It took some time for the nightingale. So here we really have an interspecies communication and a readout for the nightingale, what it is singing in response. Next. So I would say the first part that we can communicate with our own vocalization and also artificially generated one, we fulfill. But what about the context? So this was all done in the breeding season in Germany. Next. And to address if we can also go to a different context and a different continent, we went to their wintering quarters, where they typically do not, or not typically, they do not breed in the winter, but they still have to defend their territory. Next. And repeated the same experiment again. In this case, Giacomo was whistling to a nightingale, but we also did controlled experiment. Next. And I hope you could hear, and you can also see that the nightingale responded. And this was rather surprising to us, because it has been previously thought that nightingales do not match songs in their wintering quarters, and they also do not have elaborate singing behavior. Next. And with this, we have 2 different contexts where we can communicate with the nightingales. And now the remaining question would be, can we also put some meaning to the nightingale songs? And with this, I would like to hand over to Jan, who will explain you how we addressed this part.
Thank you, Daniela. So Daniela told you how we can interact vocally with the nightingales in this very precise and quantitative manner using the whistles. However, most of us find nightingales fascinating not because of the whistles, but because of their beautiful and complex songs. So to infer the meaning of these songs, we needed tools to quantify the bird's responses, specifically indicators of aggression and arousal. Next. To achieve this, we equipped some nightingales with accelerometers, and we also used thermal cameras to track their movements. Next. Next. So with the tracking in place, we could now present the birds with different acoustic scenarios and read out their behavior. First, we played back the song of another nightingale, which is a common territorial interaction. And then we also played back recordings of the individual bird's own song. And just as you feel weird when you hear your own voice, we thought that this might elicit a strong reaction. Next. And indeed, our accelerometer data showed that birds moved more when hearing their own voice compared to their neighbor's voice. This suggests a heightened state of arousal and potentially aggression, and thus a different meaning for the different types of songs. Next. Next. This heightened response to their own song prompted us to analyze the songs produced during these interactions. Next. And we discovered that the birds matched specific songs more frequently in this aggressive on-voice context. And based on this correlation, we call these preferentially matched songs aggressive songs. Next slide. So far, we've looked at vocal interactions between males. Next. But how about another context, interactions with a female? Next. 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 And while there are syllables used in both contexts, there are also syllables that only occur in the presence of a female, never during interactions with a male. And we call these unique vocalizations love syllables because we think there are song components dedicated to courtship and mate attraction. So by observing the context in which different songs are used and how they're responded to, we can assign meaning to specific elements of the nightingale's beautiful and complex repertoire. Next. One step back, please. Oh, I think there's a slide missing. Okay, so to wrap up, these are our first steps towards communicating with nightingales. We've interacted with the birds using their own signals, using the whistles and complex songs. We did so in different contexts. On the one hand, during different periods, the breeding season in Germany and the winter quarter in Africa, and with different partners, male rivals and female mates. And with this, we're beginning to infer the meaning of their song. Next. So with this, I want to thank everyone involved in the study. Special thanks to Giacomo, Sebastian, Jonathan, Susanna, Carolina, Niels, and Mira, and the entire teams within both of our labs. And finally, we'd like to thank the Coller-Dolittle Foundation for the invitation to present our work. Thank you.
Thank you, Jan and Daniela, if you can join us again. And I remind the audience that you can present your questions in the Q&A. But let me start with maybe a fundamental question. You presented beautiful data about the ability of these birds to reply to the whistles of, was it Dr. What was his name? I forget. Dr. Dan, is it?
Dr. John.
Dr. John. I wonder, by the way, if it's a prerequisite to know how to whistle to work in the project.
That's why he was hired.
Exactly. But why do you consider this communication rather than merely imitation?
Because I mean, the whistling is a kind of a long-range communication signal. So they want to convey something that broadcasts far away. And the imitation, and of course it is an imitation, but yeah, I consider it that there's the goal to match the whistle, which is also is also a sign of fitness, because if they can basically showcase that they can sing what you just sang to them, then it's kind of good to attract the female as well.
Yeah, maybe to add to that, we think these whistles are basically tags. So you have many birds singing all the time. And so if you basically repeat or start your response with the same pitch as the other bird, and you address that bird, and you can you make clear that you're responding to a specific bird and not just anyone.
So if, for example, in another scenario, they would reply consistently with a specific song or whistle that is not identical to the one presented by Dr. John, but is consistent, wouldn't you consider this a higher form of communication because there is some signal messaging?
Jan, you want to?
Okay.
Okay. So I guess what you're implying is if there was, I think I didn't get the question right, because so you're implying that if there was a different vocalization that is a messaging.
Yeah, instead of repeating the whistle, if they would have replied in a different whistle, but it was consistent, so then we could, like a conversation, I say something.
Yeah, I see what you're saying. So I think this is indeed the case. So it's hidden in the data, but when we did this frequency matching versus duration matching, sometimes they choose the one over the other. So depending, we don't know what it depends on, but it seems as if there is a kind of choosing from their vocal dictionary what kind of song to use. And yeah, you're right.
We have a question from one of our judges, Elodie Briffer, and she's asking, is there any minimum frequency below which or a maximum frequency above which they either do not consider the whistle as conspecific sounds anymore and hence would not respond or would not imitate?
Yeah, so we probe them in a frequency range between 1,000 and 8,000 hertz in steps of 200, and all of these frequencies work. If you go below or higher, then they wouldn't consider them to be, I mean, I don't know if they don't consider them to be conspecific or if they are just not able to produce them because they are not producing these sounds. And also the hearing range of nightingales is only up to 10, 11 kilohertz. So above that, they wouldn't even hear it.
And maybe one last question. So with humans, recently we see more and more studies about individual differences. So not only looking into what is common to all the participants, but also what differentiates between them. Do you see differences? For example, do different male nightingales sing different songs, or is it all unified across the species?
Yeah, so different or individual birds have 200 to 300 different songs, and part of that repertoire is actually shared. So roughly 50% of the songs are shared between all the males, and 50% are individual specific. So they can basically choose what to use for communication.
Interesting. Thank you so much. This has been fascinating. And again, all these projects are just inspiring. I don't envy the judges for having to choose one of them. Thank you so much, Daniela and Jan. Thank you. We are moving to our third presenter, who is Sophie Cohen-Bodenes, a cognitive neuroscientist from École Normale Supérieure, who will present her team's research, Cuttlefish Interaction, Arm Wave Signs, co-authored by Peter Neri, principal researcher, also the École Normale Supérieure and Istituto Italiano di Tecnologia. Sophie.
So good morning, everybody. It's a real honor to be here to present our study entitled Cuttlefish Interact with Multimodal Arm Wave Signs that I conducted during my PhD under the supervision of Peter Neri, that is also here and will be present during the question and answer. So we can go to the next slide. Yeah, thanks. So for this project, we dived into the wonderful world of cuttlefish communications. So cuttlefish are wonderful animals with a unique ability in the animal kingdom. They can do dynamic skin patterning for camouflaging. They have sort of a neural network that you can see at the naked eye. And what's even more fascinating is that this ability is versatile and that cuttlefish also use patterning to communicate. So for example, on the right video, you have a cuttlefish that switches from white to black when she's upset. So on the next slide, thanks. We will see that cuttlefish communication is also multimodal, which means that it involves several perceptual modalities. For example, when cuttlefish are mating, you can see that a female cuttlefish will release ink, which is a chemical form of communication to show that she disapproves of the act of mating. And it's also very well known in cephalopod literature that cephalopod and cuttlefish communicate with various body and arm postures to communicate, like the extension of the forearm along with zebra patterns, like you can see on this video, for male cuttlefish to express dominance. Next, please. So what we uncovered is previously undercommunicated communication signal in two species of cuttlefish, Sepia officinalis and Sepia bandensis. So as you can see on the video, cuttlefish display very stereotyped. We observe that they display very stereotyped movement of the arm, extension of the arm, long-lasting, very specific patterns that are also repeated into sequences and also displayed along with specific coloration patterns. So we hypothesized that those signals might be a communication signal. And on the next slide, I will present you the four different types of signs that we identified. So when doing the up sign, the cuttlefish is extending the arm on the upright vertical direction. When doing the side sign, the cuttlefish unroll the arms on one side or the other of the body. And it's very interesting because those signs also possess a form of laterality. When doing the wall sign, so I don't know if we can play the video, but actually during the wall sign, the cuttlefish unrolls the arm completely beneath the body, and it changes the shape of the head, making the eye look more prominent. And at some point we were wondering that it was maybe to mimic some form of octopus. So we tested this hypothesis, but our preliminary result pretty much discard this, and we think it's more complex communication signals. And also, if we can please display again the crown sign video, you will see that when the cuttlefish is doing the crown sign, it also displays very vivid bright skin patterns, for example, orange skin patterns that are communication skin colors along with black spots. So we wanted to test the hypothesis that those displays are communication signals. So how did we do it? So we will see this on the next slide. So we wanted to find a good test for that, to show that there is some social information in those signals. So a good test from that comes from the literature in human vision, and it's a paradigm that is called the inversion effect. So here I'm showing you an example with the Thatcher illusion. When I'm presenting you the two faces like this of Margaret Thatcher upside down, you have the impression that it is actually the same faces. But actually, the faces have been manipulated. And when the faces are lifted on the normal direction, you notice something that you didn't previously notice, that is, there is a gross distortion of the mouth and the eyes. And why we do not see it when the faces are upside down? It's because our visual system is very tuned to, for evolutionary purposes, to recognize faces. And so we adapted this test, this inversion effect test, to cuttlefish to see if there are communication signals, and we will see this in the next slide. So what we did is that we sampled many examples of cuttlefish signing, different types of signs, and we did a first visual experiment, non-invasive behavioral experiment. We presented to the cuttlefish just on the screen a normal version of cuttlefish signing and the control upside down version. And we were first very amazed to see that the cuttlefish was curious and interested about the display and waved back at the display. And on the next slide, we will see how we quantified those results. So indeed, so we did qualitative, quantitative statistical analysis that I'm presenting you for two species of cuttlefish. So on the y-axis, I'm showing you the frequency of signing when displaying the sign, and the red plot corresponds to signing to the upright configuration, where blue plot corresponds to the upside down. And you can see indeed that the effect was much stronger for the upright direction. So how did we interpret this result? First, we believe that we found a new method of visual playback to elicit the sign and to be able to have a way to make them elicit the sign. We also believe that it shows that they perceive the sign visually. And finally, because we replicated the inversion effect, we also believe that there is some information that shows that it's possibly communication signals. And on the next slide, I will present you a second experiment that we did. So also, we did other qualitative observations, and we saw that when cuttlefish are also visually occluded, they still display the sign. So what was our hypothesis is that, well, we state that when cuttlefish sign, they display specific waves in the water, patterns of waves that could be perceived by two candidate organs for the perception of low frequency vibrations in the water, which are hair cells on the arm, which is the lateral line organ, or the statocysts, which are akin to the ear invertebrates, because it's very important to notice that cuttlefish do not have ears, so they cannot vocalize like dolphins or like whales, but instead, we believe that those signs can be used to display vibration signal, especially in the dark, where visual modality is not involved. So how did we test this? We will see this in the next slide. So we designed a playback experiment with a hydrophone. So it consists in, so we had the cuttlefish spontaneously signing, and we recorded with a hydrophone the trace of that sign in a normal configuration. We analyzed the spectrogram, and indeed we found an expected power at low frequencies between 15 and 25 hertz. Then we constructed controls. So we reversed the trace, which is equivalent in doing the Margaret Thatcher illusion for speech. And why this control is interesting is because if I were to play back you speech reversely, it would become intelligible, but it would still preserve the low-level features of the speech. And we did a second control, which is a scrambled version, where we completely disrupted the phase. And by disrupting the phase, we disrupt also the meaning of the sign. And on the next slide, we will discover the response of the cuttlefish to this playback. This was completely amazing because the cuttlefish were not constrained, but by themselves they were coming to the hydrophone, and they would display to the hydrophone without any visual stimulation all the different combination of signs, also with vivid skin patterning. So on the next slide, we will see how we quantified this effect. And so indeed, so we find that they responded more for the normal version rather than for the reverse and scrambled version. And also for Sapia magnesis, we used a different method, so also to have more proof by using a speaker. And we also showed that they display more frequently vivid skin patterning during the playback, normal display playback. So on the next slide, I will try to convince you why cuttlefish are a very good model for the Coller-Dolittle challenge. Because we want to decode the meaning of the sign to crack the cuttlefish communication algorithm. And what is the actual challenge here? Where we know that cuttlefish do skin patterning for communication with black spots, with very elementary features like stripes, like squares, that can be very well captured by a computer vision algorithm and principal component algorithm. Furthermore, we have now given another type of communication, which are the arm wave. But what we want to try to understand here is why, so on the first video, why sometimes cuttlefish will do orange and two black spots, but why sometimes it's just in some other behavioral context, it's just becoming orange, and why in some other context, it will become bright orange to the crown sign and do 3 black spots. So we made first development to crack this cuttlefish algorithm that we will see in our next slide, because we released in our previous paper and developed an artificial intelligence algorithm that enabled us to encode all those little features of skin patterns and to decode them with machine learning algorithm to analyze the dynamic in time series of those skin patterns. Currently, we are augmenting this algorithm to categorize automatically the different wave signs, and we are collecting large data sets, because it's only by collecting large data sets and making the cuttlefish elicit the sign in various behavioral conditions that at some point we will be able to extract the patterns in those and see which specific decorrelate and see which components corresponds to what. And on the next slide, I will also show you something also surprising, is that we also believe, so as you can see here, this is not a cuttlefish, this is another species cephalopod, it is a little squid that is displaying the role sign to a shrimp. So shrimp is, so it means that we believe that also the sign can be not only an interspecies communication signal, but intraspecies communication signal that can be displayed not only from cuttlefish to cuttlefish, but from a cephalopod to other cephalopods, and also from cephalopods to prey. And we also saw cuttlefish displaying the signs to us. So we believe they are a good candidate for the challenge. And finally, on the next slide, I will show you what is our next goal. So our next goal is currently, so we have designed a prototype of the Coller-GPT. So what is a Coller-GPT? Is what we would like to develop is an underwater robot that would enable us to communicate finally, possibly with the cuttlefish. So this robot would be a type of hand gesture robot that would be equipped with hydrogel tentacles, so softly actuated tentacle that could actually very mimic the sign in the water because they can bend softly, and it would really better reproduce the waves in the water and the vortex, etc. We would have this algorithm to recognize automatically the sign, and then it could send the signal in a loop back to the robot, and the robot would display the sign, and it would help us to have more insight on the meaning of those signs. And finally, something that is very important for us and that we really want to push forward is that we have developed non-behavioral, non-invasive experiments, and we really want to push forward those computational neuroethology tools, those robotic tools, because now their technology enable us to perform experiments that are non-invasive, and we want to be able to refine experiments with those animals that have proven to be so clever and wonderful. So we thank you very much. On the next slide, we want to show you a nice picture of our cuttlefish. We thank everybody who supported this project, and of course, we thank our wonderful animals. Thank you for your attention.
Thank you very much, Sophie. That was really fascinating. There are several questions, both from the audience and from our judges. But let me start maybe with a question of my own about the first experiment. So I think it was brilliant to do the inversion effect on the cuttlefish. But how can you tell that this is specific for social communication? So if you would have performed ask them or kind of push them to perform another task that does not involve social communication, do you still, maybe they would still show a difference just because they are surprised by the upside down face? Do you have a way to show that it's specifically for communication?
This is an excellent question. Actually, this is very something that is at the core of our what we are wondering and that we are trying to do and that we are trying to prove, because it's very difficult to say if, for example, the cuttlefish is just not responding to some movement, you know. So we are trying currently really to find a good stimulus that would tell us that there is some social content, some social information. So that's why we use this upside down effect, but also currently, so we have preliminary data that cuttlefish also respond to videos of praise. So we are we would like to generalize maybe this paradigm using, for example, biological motion, point-like displays to really show this specificity that it's responding to something biological and not only something that is just some movement that attracts them.
Yep, thank you. Another question from our judges, again from Elodie. So you played videos of themselves. Why did you choose that option instead of a video of another individual, which would have been more neutral?
Oh, no, okay. So it's a very good question. Maybe it's because I was not very clear. So we did actually both. So it's not videos of themselves as if they were looking at themselves in a mirror. It's videos of other cuttlefish. So they are looking at different type of cuttlefish. So, but it's not a reflection of themselves. For example, we showed other males, we showed other females doing different type of sign in different conditions because we wanted to see that, and also we wanted to see if there is, for example, they would do specific sign when they see a male, specific sign when they see a female, or this type of things. Yeah.
Thank you very much. There are so many questions, but unfortunately, I'm getting signals that we are out of time and we must move to our next presenter. So thank you again, Sophie, for this talk. And last but not least, we are going to hear now from David Omer and Guy Oren, both from the Edmond and Lily Safra Center of Brain Science at the Hebrew University in Jerusalem, and they will present their research, Naming Behavior in Marmoset Monkeys, Offering Clues to Human Language Evolution.
Okay, so thank you very much. So we are going to present our latest study, vocal labeling of others by non-human primates. Yes, can click again, thank you. So first of all, what is vocal labeling? It is the ability to assign a unique vocalization to an object or a conspecific. So human naming of others is a sub-phenomenon of vocal labeling and an important part of human language. It allows us to increase the sophistication of the way we cooperate with each other to perform social behaviors. And it involves vocal learning, which is the process of learning a new vocalization or a new use for an existing vocalization from others. Next. Thank you. Besides humans, very few species are known to vocally label their conspecifics. So, of course, dolphins, as we've heard. But also African elephants have recently been shown to vocally label their conspecifics. However, so far, there has been no evidence for vocal labeling of others in non-human primates. And that is what we set out to investigate. Next, click. Thank you. We studied marmoset monkeys, which are small social New World monkeys with a complex vocalization system. They are about the size of a big rat, and in nature, they live in small family groups in the rainforests of South America. They have a large repertoire of different vocalizations that they can make, and we focused on a specific one called the phi call, which is a high-pitched call that sounds like this. As we can see, it is monotonously increasing through time. But a phi call is not just a vocalization, it's also part of a unique marmoset behavior. Whenever two marmosets lose sight of each other, for example, in the foliated rainforests where they live, they immediately start exchanging phi calls in a turn-taking manner, as shown in the example in the bottom right graph here. Next. So in our lab, we were able to record phi call dialogues by placing two monkeys in the same room, as shown in the cartoon on the left. And as soon as we place a visual barrier between the monkeys, they start to engage in natural phi call dialogues. For example, the one on the right, which we recorded in our lab. Next. So using those recordings, we constructed a huge database of over 50,000 calls, including 10 different monkeys who belong to 3 separate family groups. And each call was fully labeled, including who the caller and the receiver were. Next. To analyze the calls, we embedded each one in 80-dimensional space by concatenating the normalized frequency and amplitude modulation vectors. We used 40 dimensions for the amplitude modulation and 40 for the frequency modulation time course. And now David is going to tell you about our results.
Thank you, Guy. Can you hear me?
Yes, we can.
Okay, we use machine learning classifiers to test if calls from a single monkey contain information about the receiver identity, by testing the accuracy of the classifiers to detect the true receiver identity. On the left is example from one monkey, Adonis. You can see the accuracy of the classifier to detect the identity of each of the receivers compared to chance level when we shuffle the receiver identities in gray. classifier's accuracy was way above chance level for all the monkeys in our experiment, and as shown on the right, by the highly significant accuracy distribution compared to the shape distribution. So receiver identity is encoded in fecals, but do monkeys perceive and utilize this information? To test this, we conducted a playback experiment where we, instead of placing two monkeys in the room, we placed one monkey and a playback system on the other side of the visual barrier, and had a monkey engage in fecal dialogue with the computer system as shown on the left. When we tested the probability for a real monkey to answer the coded call that originally was directed to him against the probability to answer a non-directed call, we found that it was significantly more probable to answer directed calls versus non-directed calls, which shows that not only the receiver identity is encoded in fecals, monkey also perceive this information and respond correctly. Next, we asked if different monkeys use similar calls to address the same receivers. To do this, we calculated the similarity between calls of two monkeys when they were calling the same monkey. Projecting these average similarities onto Euclidean space, as you can see here on the left, show that the monkey families are clustered together, suggesting that monkeys from the same family tend to use similar calls to address different monkeys. Further, the similarity between calls from monkeys from the same family was significantly more similar when they were calling the same monkey than different monkeys, showing that there is a similarity component which is specific to the identity information in the call. The clustering into family groups also revealed evidence for vocal learning in the adult marmoset monkey, because members of family groups A and B, for example, are not genetically related and were paired as adults. So the only way they could use similar names is by learning those names from other family members. Finally, we asked what are the specific acoustic features that are used by marmoset monkeys to encode the receiver information. We defined a set of acoustic features and tested the explained variants of each feature. At first glance, as you can see here on the left, no specific common set of features seem to be used, but when sorting according to family groups, again, as shown here on the left, each family used a unique set of acoustic features to produce fecals, and when projecting again to Euclidean space, family groups were clustered together, showing that different families use different dialects to encode the identity of their receivers. The importance of these findings lie in the context of human language evolution. Non-human primates' vocal communication, until very recently, was considered innate and non-flexible, and therefore evolutionarily non-relevant for you understanding the evolution of human language. Our results show the contrary. So where do we go from here? Our next step is to harness the state-of-the-art machine learning tools to study vocal communication in marmosets. We are constructing a large language model of marmoset vocal communication using a huge recorded data set that includes spontaneous recording from the colony. It includes all the large repertoire of marmoset calls. We tend to use the fecal conversation, which are fully labeled, to test the validity of the model, and use the model to learn about the syntax of vocal communication in marmosets. Here are all the members of the Omer Lab and the funding agency, and I'd like to thank you for your attention. I'll be happy to take any questions.
Thank you so much. This was really outstanding. I'm starting to run out of compliments, but I swear that they're all genuine. So maybe as a first question, just to understand these labels, are they personal names like the ones we have for humans, or are they labels marking some role? Do we know if they have semantic meaning?
So actually, the naive, the obvious answer is that we don't know. And this is why we deliberately use the concept of vocal labeling instead of naming. So what we definitely see here that a marmoset monkey vocally label other conspecific, whether this is names or other labels, it's not really clear at the moment. That's it.
Okay, thank you. We have a question from our judges, quoting, I'm hoping that I'm pronouncing the name correctly, Kelly Jekola, who published a paper suggesting an alternative explanation to your result. And the main point, if I understand correctly from the question, is that you did not show that these calls are indeed labels, so that they represent specific individuals in the minds of the marmosets. And basically the question was, are you planning to run such a test in the future to see how individual it is, or use a cross-model setup?
I'm not sure I understand the exact question. So she asked whether
She asked whether what you've shown is that these are labels that represent specific individuals in the minds of the marmosets, and suggested that maybe there should be a cross-model setup in which this would be demonstrated, and she asked if you are planning to run such a test in the future.
Well, I think we have a very convincing evidence that the labels are actually vocal labels. Actually, it's very similar to the previous questions. We cannot definitely say that these are names or similar to naming in humans, but the labels are definitely specific to individual conspecifics. So I think in that sense, we have a good answer to this.
So maybe one last question, do we know how they are acquired? Is this something that is passed through individuals? Is this passed through imitation?
So obviously, every such type of learning involves imitation, and in principle, there are two possible types of imitation. The first one, which is a relatively simple one, is imitating the calls of the other receiver, as is done in whistle calls in dolphins. What we show here is something a bit different. What we show here is a different type of imitation, and that's the type of a single monkey watching two other monkeys exchange vocal labels and learning to label a third monkey by imitating the calls of the previous one. So it's a different process, and which implies that it's more sophisticated than just simple imitation.
Thank you so much, David and Guy, for this presentation, and thank you to all finalists for these inspiring and highly interesting ideas, truly groundbreaking research. And now the judges panel will retire to deliberate and select the winner of this year's challenge. As I've said before, not an easy task in any way. The winner will enjoy a talk by, in the meantime, while we are waiting to hear what the results are, we will enjoy a talk by our special guest, Dr. Irene Pepperberg. Irene, can you please turn on your camera? While you are doing so, I will say that Dr. Pepperberg is an adjunct research professor at Boston University, and we are truly delighted to have her with us to talk about her studies on the communicative and cognitive abilities of gray parrots. Irene, are you with us?
I'm trying to start my video.
Oh, here you are. We can see you and hear you.
And my screen. Okay. Share.
And I'll just remind, in the meantime, I'll just remind the audience that you can, again, upload your questions in the Q&A, and I will convey them later on to Dr. Pepper.
So it says I'm screen sharing, but the problem is, okay, there we go. I want to get to slideshow from beginning. Yay. Okay. Everybody can see it's working?
Yes. We are well prepared for your talk. Thank you so
much for this honor of keynoting this. It's quite exciting. Okay. So my first slide is the usual. Thank you everybody for all the help over the years. Okay. But the main thing is, why should we train non-humans to use a human-based symbolic system, all right? Which was the original way of looking at interspecies communication. Don Griffin in '76 came out with a book, and this was after some of the work was started, but he said we can use it for direct questioning. We could provide a possible window on the minds of animals, ask them what they're doing and why they're doing it, for reasons like that. And it would allow us to explore cognitive and possibly linguistic capacities of non-humans. And Ewan MacPhail argued that language was necessary for any kind of advanced problem solving, but he argued that only humans could achieve anything that was even language-like. David Premack, who was actually examining language-like behaviors in non-human primates and chimpanzees, said that they could learn some level of symbolic reference, okay? And he also argued, and this was really important, that acquisition of reference actually changes how they process information. So he was actively interfering, in a sense, with their natural systems. So his argument that such behavior would be limited to our closest relatives would seem to make sense, okay? But our closest relatives are not vocal learners. And what I'm saying by vocal learning is the extent of vocal learning, learning totally new systems and new communication systems and new languages, okay? They lack the flexibility that would present open-ended learners like humans, okay? And this is important not only to refer to what's known, but respect to novelty. So as humans, we can come up with new words like glitzy, okay, and airhead. We can come up with new words to describe an entire new area, bits and bytes and rams and things like that, that we never would have had before. And species that were vocal learners, the one most frequently studied, the songbirds and the parrots, they were separated by humans by 300 million years of evolution. Even though they had things like motherese and basically babbling periods and things like that, we're talking 300 million years. So when I argued in the early 1970s that I wanted to study animal-human communication using a parrot, my first grant proposal literally came back asking me what I was smoking. I wanted to work with an animal separated by 300 million years of evolution from humans, unlike the great apes, and I wanted to use an animal that had a brain the size of a shelled walnut, unlike animals like the dolphins whose brains were enormous. And there was some really good other reasons to denigrate my work because it had been tried before. People like Maurer and Greislein and Zahner had used the standard operant training of the day, and they had failed horribly. In the operant system, for those of you unfamiliar, you look at the animal's brain as a black box. You starve it down to about 85% of its normal body weight, so it will be very hungry and work for a food reward. You present it with a stimulus. You hope that it will come up with some relevant response to that stimulus. If it is, you give them a piece of food. It did not work. Why? Well, let's look at an example. Maurer was trying to train a bunch of mimetic birds to say hello when he walked into a room. That was very easy. They were hungry. They learned quickly to say hello because they got fed, but they were still hungry. So they kept saying hello, 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 in all sorts of inappropriate times. They weren't reinforced for those vocalizations. The behavior became extinguished and there was failure to learn. Greislein and Zahner locked a bunch of minor birds in sound isolation boxes, played them tapes of human speech. The birds learned nothing from those tapes, but learned sounds in the laboratory, things like dripping faucets and creaky doors. So the idea was that these birds are, mindless mimics, absolutely incapable of doing anything at all intelligent. Then 1975, Dietmar Todt has a paper in Zeitschrift showing, hey, guess what? We can do this. And Todt was a junior professor in the lab of Otto Koehler. Koehler was interested in number concepts in birds. We're going to talk about that very briefly later. But he was also interested in what he was hearing, what was going on all these animal-human communication systems. And he knew that these parrots were mimetic. So he hired Dietmar, who had been studying song learning in birds, to train a bunch of gray parrots to communicate with humans using human speech. What Dietmar said, hey, birds, how do birds learn in the wild? Okay, the baby bird is sitting in the nest. He watches dad sing one song and it defers all the, gets all the other birds that are trying to invade our territory away. And he sings another song and mom may come in for food and things like that. So the bird learns by observation what's going on. So the brain is not a black box, but it's a social information processor. And he developed a model rival technique to train these birds. And I don't have time to get into that. Maybe it can come up in the questions or you can read some of the papers. But the bottom line was, you had a bird, okay, that was learning this, but there was still the question. the brain. Okay, when I would present my studies, people would say, but this is impossible. There's no cerebral cortex. And if you look at the bird brain, it's a blob of protoplasm. There's no sulci, there's no gyri. The relative size is, it's the size of a shelled walnut compared to our brain. How are they doing this? And I would tell people when they argued with me, I'd say, hey, you're the neuroscience, go figure it out. And in the 1970s, almost nothing was known about avian brains. People like Carton were beginning to look at this stuff very seriously, but it wasn't until 2005, Nature Reviews Neuroscience, Jarvis et al. Were 20 other people that they actually isolated this equivalent functional cortical area in the avian brain. In 2005, there was also a wonderful paper that showed the relative size of this brain compared to the rest of the brain, okay, was enormous, and the same relationship in the parrots and the non-human primates. So here's the non-human primates, there's the parrots, and they're on the same line, the body mass versus brain. 2015, okay, we knew before that there were 7 structures in the avian brain and 7 structures in the human brain that were related to vocal learning. But in 2015, they found another area of the parrot brain that was specifically used for heterospecific learning, and the relative thickness and density of this area had to do with the amount of heterospecific learning the birds could do. So gray parrots had tons of it, which are very great mimics. Kias, which learn their vocalizations, but not much heterospecific learning, very small area. Okay, 2016 showed the neural density in the parrot and corvid brains were much higher than that for primates of equivalent weight. It was actually equivalent to that of the larger non-human primates, the apes, the great apes. In 2020, a paper showed that the connections among various areas of the avian brain appeared homologous to those in primate brains, even if they weren't in the same spots, and they were looking at animals as diverse as owls and pigeons. Another science paper in the same issue claimed that if you believe that you have found the neural correlate of consciousness in humans, they had found the similar thing in corvid and probably in parrot brains. And they were talking to homologies. 3 new papers very recently came out, however, saying, no, it's not homologous, it's analogous. That both brains started out from the dinosaurs, evolved in separate ways, but basically ended up doing the same things through evolutionary pressures. But these studies are based on chickens, which are not vocal learners, and the chicken branch, what led to the parrots, corvids, and falcons closely related to parrots, branched off very, very early. So maybe there's something very specific about the chickens that are not homologous. We don't know. This is for us to study. The bottom line behind all of this is that after studying gray parrots for, at that point, 30 years, okay, I found that, yes, they could learn a certain amount of English speech and communicate with us. Alex had labels for about 100 to 150 different objects. He had labels for 7 colors and 5 shapes. Importantly, had labels for categories. So he didn't know just what was or was not green or three-cornered, but that green and three-cornered were subsumed under hierarchical label of color and shape, and he also had one for materials, for things like paper and wood and things like that. And once you had concept of category, you could start looking at things like concepts of same and different, which I will talk about in a little bit detail, concepts of absence, relative size, number. He had absolute number concepts, not approximate, but absolute. He learned through joint attention. He had a very limited level of recursion, not the linguistic one is this is the cat that bit the rat that ate the cheese and they all lived in the house that Jack built, referring back to the house that Jack built. But you could show him a tray of 7 different objects and say what object is green and 3 corners, or what shape is green and wood. So it could refer back to the original question. And there was a conjunction of the 2 attributes there. He transferred his concepts from one type of task to another. So when he learned absence in terms of same and different, saying nothing was same or different by saying none, he transferred this to the absence of relative size by using none without any training, and to a zero-like concept with numbers without any training whatsoever. He learned through a fast mapping of a sort, a little bit different from the way children do, but he asked questions, which was the only animal-trained, non-human, animal-human communication. He asked questions. He asked his color when he saw himself in a mirror. he was told he was gray, and then he did riffs on that. So he went gray, grape, chain, cane. And if we could give him objects that mapped onto those objects, they became embedded in his repertoire immediately. And here's a little bit of vocalizations. You can hear them.
I'm sorry.
No contrition, but he knew appropriate times to say it.
What color? Orange.
Okay, so, but what exactly is the symbolic reference that I'm saying is so incredibly important? Well, it's one aspect of human language. And if an animal has that, they have the beginnings. It doesn't mean they have language like you and I have it, okay? But it's critical to all else that follows. And according to Deacon, and now this was in 1997, before we knew so much what more we know now, he said it was a central problem of language origins and likely separates human and non-human communication systems. Okay, we don't think that's true anymore, but this was the theory on which things were based at that point. But once reference is established, information content of the symbol can be manipulated independently of physical instantiation. And this is what Premack was getting at. An example is mathematical symbols. You have 3 blocks, you have 3 hearts, and you can talk about 6 things. It doesn't matter what things they are, okay? It does not guarantee, but enables abstract thought. You can represent the object, the action, the attribute by a symbol that you can mentally manipulate, okay, releasing the thought processes from the here and now, okay? So you can plan your actions, you can alter them without being physically embodied. The joking way of looking at it is your hypothesis dies and not you. Okay, so what evidence do we have that symbolic representation affects cognitive processing? Well, I'm briefly going to review all these different things. Again, it's going to be lightning fast because I don't have a whole lot of time. But the point is, in the second half of the 20th century, a number of researchers attempted to train non-humans to acquire human-based communication code. So the Gardners, Penny Patterson, Lynn Miles all used American Sign Language or variations thereof. Premack used a bunch of plastic chips, so each chip represented a label for an attribute or a label or a person's name or things like that. The Rumbaus used a computer system, okay, where their animals, it's called Yorkish, they had various different symbols. Lou Herman initially he used an auditory system, but he went into a sign language. Schusterman used signed and symbols. Diana Reese used a computer-based system, and I used vocalizations with Alex. And we all got some level of success, okay? They did learn symbolic representation, though not communication at the level of human language. None of these animals could communicate the way you and I are communicating. They were basically at the level of very young children who were beginning to acquire some levels of communication systems. And those that did acquire such representation also acquired specific types of cognitive abilities, okay? And one of these things was same and different. And why is same and different important? Because it's a complex concept. It's not simply identity versus non-identity or differences in entropy, okay? According to Premack, if you're doing same different judgments for 2 items, you have to do a feature analysis of the objects being compared, recognize, and this is really important, that they can simultaneously exhibit attributes that involve both similarity and difference. And you have to understand which attributes are being targeted based on what kind of question. Are you being asked about similarity or are you asked about difference? Okay. And here they're different shape, different color, but presumably the same material. So if the subject understands this, they have to attend to multiple aspects of 2 different objects, determine from a verbal question, okay, whether you're asking, is it sameness or difference? Determine what exactly is same or difference, okay? And then produce verbally the label for the hierarchical category of the appropriate attribute. Not simply say same, different, okay, which is identity versus non-identity, but tell you what is same and difference. And this is exactly what Premack was getting at. Only those animals that had symbolic reference could do this type of task. His apes without symbolic reference failed miserably. So I was working with Alex, a gray parrot, who had some symbolic reference, and he succeeded in this task. He worked including sets of objects and attributes for which he had no label. We went to a Toys R Us and got random things he had never seen before, and we still asked him what was same and different. He responded statistically significantly to same and different, with the colors color, label, shape, or mama. The t was a little bit hard for him to say. And if nothing was same or different, he learned to say none. Again, he was not simply saying same or different. He had to produce the hierarchical category label, and at that time he had about 70 different labels in his repertoire. Again, only apes with this kind of ability could succeed. So he performed at the level of apes, and also like 4-year-old children who were trained on 2-item array-based match-a-sample tasks. He had exact number concepts, okay, not approximate number, but exact. So he could look at a tray of this with red and blue woods and wools, okay, and tell you how many blue wool versus rose wool or woods and things like that. He could add small sets of number of objects and of Arabic numerals. He learned the Arabic numerals that represented these sets, okay? Again, he had comprehension, and comprehension is important. You could look at take a 4-year-old child and say, how many marbles? And they'll go 1, 2, 3, 4. There's 4 marbles. And you say, ooh, this child can count. Then you give the child a bowl of marbles and say, give me 4. And the 4-year-old picks up a whole bunch and hands you the entire bunch. They don't have comprehension. But we could give comprehension with Alex, and he could look at a tray like that and respond to what color 6, okay? He could infer cardinally from ordinarily, meaning after he had learned his number line, okay, he knew that the next number was exactly one more than the previous number, the successor function. So when we taught him a new number on the number line without making it refer to a set, he could infer that that set meant one more object. We studied with Griffin, another bird, we studied Piagetian probability, something that 5- or 6-year-olds get. Okay, what do I mean by that? Okay, you take a bucket, you put in all these different objects, okay, you shake it up, and then you remove one and you hold it in your hand and you ask the child, which color do you think this is? If the child is about 4 years old, they'll say something like, oh, red's my favorite color, so it's red. Or they'll say something with maybe a little bit more thought, but not exactly right. Oh, you put one red in and you took one thing out, so it has to be red. Only when they're about 5 or 6 do they say, oh, wait a minute, it's a probability. There is more blue, so it's more probable that the thing in your hand is blue. Griffin correctly tracked the probability for multiple sets of different objects, okay? So it was freaky that he did it completely right. We studied optical illusions from children through adults. So Alex looked at this Mueller-Lyer illusion, and those two horizontal lines are the same size, but he saw one is bigger than the other, just as you see it in this thing, and he could tell us what color was bigger or smaller. With Griffin, we looked at Kanitsa objects, and it's hard to see, but there's a hexagon there, okay, and occluded objects. So if you give this to a pigeon tells you this is 5, the green thing is 5 corner, essentially. Griffin could label it as 4 corner. Other tests did not use productively directly, but believe we were assisted by their understanding of symbolic representation. So we studied Piagetian liquid conservation. Again, this is something that kids have to be about 5 years old to fully understand. You give a child 2 glasses half full with juice and you say, which one do you want? And they giggle and they say, it doesn't matter. Then you pour them into one into a tall, thin one into a short, fat one. And you ask a 4-year-old, which one do you want? And they'll point to one of them and say that one. You say, why? Because there's more. And you say, but you just told me it was the same. No, now there's more. If you ask a 5-year-old something like that, they'll point to one and they'll say, I want that one. You say, why? And they'll make a reasonable sense. Well, it looks like it's more fun to drink out of. Okay. So we did this with our birds. We also did an overconservation study to compare them directly with apes. Okay. We had 2 different sized quantities, and then we poured them into 2 cups that had fake bottoms. Okay. And we put our hands over and across them so they couldn't track just where we were putting things. And Griffin and another bird, Athena, and Pepper and Franco, another 2 birds, tracked the exact correct amounts. Okay. We studied delayed gratification, which Michelle studied in 4-year-olds, the marshmallow test, okay, where he gives the children one marshmallow, say, I'll be back in 15 minutes. If you don't eat that one, you could get 2. Okay, Griffin and Pepper, another bird we work with, would wait for 15 minutes or more for more treats. Griffin would wait up to 15 minutes for a better treat. And the killer was he did exactly the same distractive behaviors as those children who succeeded. So he would, fall asleep. He'd turn around and he'd start preening. He'd start talking to himself. He'd sometimes throw the first one off the tray so he didn't have to look at it. And the same types of behaviors. And these are his results for the waiting for more. Okay, visual working memory manipulation. This is basically a shell game on steroids. You take 4 different colored pom-poms, you cover them with little cups, you start doing bunches of switches, and only then do you say, now you find the yellow one, okay? Now, when we did it with college students, the college students had to say, 2 plus 2 is 4, 2 plus 2 is 4, while we were doing the swaps, so that they couldn't do a mental vision of what was going on, because there's one way of solving this was going like a red, green, yellow, blue. I do a swap, red, yellow, green, blue, things like that. So, but we couldn't ask Griffin to do that, okay? But he watched as I did this, okay? He beat or matched the college students up to 4 cups and 2 swaps, was close for 3 swaps, and still above chance at 4 cups and 4 swaps. And we think he might have been doing this because he knew the labels for the objects and could do this mental swap. We tested also young children, 6 to 8 years old. They completely fell apart by 3 swaps. We couldn't even ask them about 4 swaps on this thing. Griffin finally fell apart at 4 swaps, but by then he was so bored with the task, I don't think he was actually paying attention. We did a study on exclusion, the conventional 2-cup task. This is inference by exclusion. So you insert 2 different foods of equal desirability, you show it in view, you then put up a barrier, you take away 1 food, you eat it, and then you present it to the bird and you say, okay, if you want a treat, do you remember where where you can get a treat? They have to remember, okay, where did she take one out of, and where I can choose my treat, okay? And we did this with 4 birds. They were all pretty good on this. Franco had a little bit trouble in one of our other controls on this stuff, but basically they did it. Okay, so these experiments are thought to test fancy term disjunctive syllogism. It's A or B, not A, therefore B. And A and B are evaluated as a pair. Equally possible, however, is the idea that both A and B are plausible likelihoods. So if you imagine if you have a barrier up and you have 2 cups and you say, hey, look, I'm showing it, I'm going to hide it on one of these cups. You remove the barrier and then you show that A is empty. So it could, it was maybe A, maybe B, it was not A, but you don't know for sure. It was maybe A, maybe B initially. Now it's still maybe B, okay? So this is the work of Modi and Carey in 2016. And this was something all previous studies failed to entertain. So to test this idea about maybe, they did inference by exclusion, and they did it with 4 cups. So now you have 4 cups in a row. You have A or B, and you show, look, I'm hiding 1 thing under here. So there's 1 in this set. You show a C or D, I'm hiding 1. So there's 1 in this set. Then you show A is empty. So if you understand what's going on, okay, there's only 1 set here. You showed me that A is empty. It's got to be in B. And these would be only 50-50. And, you'd have to gamble. But if you're doing maybe A, maybe B on this, and you're not thinking about it in other term than maybe, you would say, well, it's still maybe B, maybe C, maybe D. And you would choose equally between those 3. Why does this seem impossible? It's not impossible because that's what 2.5-year-old children do. They can't figure out the system. 3-year-olds are getting it. They're not great. 4-year-olds are better. 5-year-olds are even better. But Griffin beat out all the kids. Okay, and there's another 3-cup task again. I don't have time to talk about that one. So, but recently, this is also fascinating. People argued that a further test is necessary in which subjects you give them all 4 things, and then you show them that A is full. They empty A, and then they have to say, oh, okay, they're given a second choice. So they have to infer that B is empty on their own, and then choose between C or D. So then they have to gamble, and they have 2 correct choices between 3 cups, so the chance is 67. And the responses have to be close to ceiling. Griffin beat out even the 5-year-olds. Okay. So to go back to Primac's initial claims, clearly symbolic representation is not limited to primates. Does it affect the way non-humans process information? We're not sure, but definitely it provides a tool that enables them to function at a maximum capability. So we can show them that these birds are really, really smart and show that these abilities are related to vocal symbolic representation. And the other birds that we've worked with also have some level of symbolic representation. So now we get to the critical point for the Coller-Dolittle game. Okay, these birds in the wild. First of all, you'd never see these many birds in the wild anymore because they're now CITES 1 endangered. But the point is, okay, my theory at the beginning of this is these birds could not do this kind of symbolic representation unless it was built on some kind of mental representation that they already had in the wild. What do they have in the wild? We don't know. This is what we need to determine. And in the wild, they have a very complex system, which would lead, evolutionary pressures would lead to needing this, okay? They lived in these flocks of 100 different birds. They had dominance hierarchies, and that was important. And they had to learn the dominance hierarchies by transitive inference. They couldn't interact with all the 100 birds. So the idea was Sam beat up on Joe and Joe beat up on me. I don't even go near Sam, you know? I can understand that, you know, this bird would beat me to bits, okay? And I also know they're extremely long-lived birds, that over the course of my lifetime, Sam might die, Joe might not even inherit, Billy and Bobby might take over. I have to have the flexibility to update all this information. The same thing in terms of foraging behavior. They forage 60 kilometers a day, okay? Huge range. And over the course of their lives, everything changes. On a daily basis, it's like, oh, we ate all the fruit on this tree today. We got to remember not to go to this tree and go to a different tree tomorrow to get some fruit. Over the course of their lives, in terms of like a nut-bearing tree, okay, this tree is dead, this one is a new one's coming up. They have to be able to change and evaluate everything that's going on. Do these nighttime, you know, resting things where there are 100 birds, are they information transferring? Was one bird say to the other, hey, I found, you know, great corn yesterday, come follow me tomorrow morning? We don't know this. This is the kind of thing that we have to study, okay? And this, again, brings up the importance of this prize. The sources of funding for understanding this thing are almost non-existent, particularly in the States right now. And we need to figure this out, okay? And we need it especially because many of these species are on the verge of extinction, and we need to learn about them before they become extinct in the wild. As I mentioned, grays, they went from birds and hundreds of birds, you know, 700 birds in a nighttime roost to maybe 100 now. People who have been in their foraging areas say, oh, we saw lots of birds. We saw a dozen, where my students in 1995 would see 50 to 100, okay? So these are really, really important. And the support of this foundation provides one of the few ways to further such research. So very quickly, I want to thank you so much. And there's probably about 2 minutes for questions, but there we go.
Indeed, thank you so much, Irene. This has been truly remarkable, and these are quite, you know, impressive birds you have on your hands. Maybe 2 short questions for just to better understand. So maybe the first one, you presented a highly impressive range of abilities, both within communication and outside of it. But perhaps the holy grail of language, of human language, is syntax. Would you say that they have some form of syntax? They definitely understand questions, it seems, but that could also be a matter of intonation. Do you think there is syntax there, some form of syntax?
Okay, our birds, and again, it could have been that the way they learned, but they would always use like adjective noun, okay? So, and the birds, they didn't necessarily learn. So Griffin did learn, Alex did learn, you know, we'd say, what's this key? What color key? Green key. But Griffin learned his color labels separately from his object labels, and he still puts them in that order. So again, he might be just listening to us and learning from us, but he certainly doesn't mess up the syntax. And the other thing that was really fascinating is when Alex would learn new labels, he'd put new labels together, okay? So he would say things like cup and paper, and he'd come up with copper, okay, for a penny. He didn't say per cup, you know, he put the phonemes in the same order. So it suggests that he seemed to learn some kind of order of what was beginnings of words and ends of words, which is a syntax-like behavior.
Very interesting. And maybe one final, more critical question about this idea. So we are all here trying to crack the non-human communication code. Can you think of disadvantages for, let's say that we succeed? Are there also downsides for that?
Well, the first one is that, I mean, bad actors are out there just using simple playbacks to get birds for poaching. And you could, you know, for birds and other animals for poaching and things like that. I mean, you could see where if you got a really complex system, you could make that infinitely worse, okay, in tracking. And the other thing we have to be careful about is what I call the Quine system. So Quine was a philosopher. The idea was if you are, let's say you're an anthropologist and you're with your, you know, somebody from a tribe who's your interpreter, and you're walking down, and this is Quine stuff, and you're walking down, you know, in the forest, and this little furry thing runs in front of you, and your interpreter says Gavagai, and you go, oh, that's the label of the species. But it's like, no, it could mean that's some little, just a furry thing that runs across. It could mean lunch, okay? There are multiple possible interpretations. And so we have to be extremely careful when we're cracking these codes, not to simply rely on correlations, on simple correlations, but to do all sorts of very kind of complex playbacks and test alternative explanations for what the meanings are of these vocalizations, or, you know, hand signals, or you know, any arm signals or things like that we're studying. You have to be really careful, not just that correlation.
Wonderful.
So,
Thank you
yeah.
so much, Dr. Pepper, both for the talk. I've learned so much, and for the words of caution that we should all bear in mind. Thank you. Thank you so much.
Oh, you're so welcome. This has been fun.
And now I'm told that the judges are back from their deliberation, and I believe we are ready to nominate the winner. So please join me on camera now, all the judges. Let's announce the winner of this competition.
So the winner is or whatever. First of all, can I just say, wow, I mean, they were all fantastic, and thank you so much for entering and sharing. But they're all amazing. But we thought that the dolphins' use of complex whistle communication, Leila Saye and Peter Tyack, were the best, and you know, could really use the money to support use of AI as they move forward. But over to Yossi as well to say more, if I may.
Yeah, so thanks, Jeremy. First of all, I want to say thank you to Jeremy and the Coller Foundation, and to all of the referees, and to Liad, of course, who really did a lot of work. And it is, as Jeremy said, it was not easy at all, but eventually the committee decided that the Dolphin project led by Leila Sayeg is the winner. We were mostly impressed by the long-term huge data set that was created, and we're sure that it will lead to many more new interesting results. We were very impressed by the attempt to perform field-controlled manipulations using, you know, sophisticated technology, speakers, drones, etc., demonstrating response and animal response in multiple contexts. And we were also very impressed by the use of non-invasive technology in order to perform the work. And as Jeremy said, we really hope that this prize can also push the use of AI in order to reveal even more impressive future results using this immense data set. Before ending, I just want to remind you that this is the first prize from 5 years. Actually, Jeremy, I noticed that you said many years when you opened, when you gave your opening talk, so maybe many years, maybe more than 5. And that we are now open. The prize is open for.
I think we're going to achieve this within 5 with the use.
Okay, so we don't need more than 5. Okay, maybe we'll have a different prize. So you can go on our website, the Coller-Dolittle website, and yeah, there it is in the chat, and submit work for the 25-26 round. I want to remind everyone that the prize is given for work already done, not for grant proposals as we usually write as scientists. Even though we try to clarify it, we got a few submissions that they were clearly a proposal for future work, and we just couldn't do anything with them. Okay, so this is for work already done. There's been a lot of amazing work, by the way, published in the past year, and I'm sure we'll have many more submissions and more impressive even submissions next year. And just one final comment: a lot of journalists and people ask me about the grand prize, the Cracking the Code 10 million prize. So I want to say that part of the reason why we started this competition is to see what is out there, what are people doing, to engage the AI community, to let them know what kind of data is available. And we're still working on the criteria for the grand prize, okay? So if you ask me what does it mean to crack the code, I just can't give you an answer at the moment. So I think, during the first year or two, we will establish the criteria and announce them for the grand prize. That's it from the scientific committee. Thanks.
Thank you so much, Yossi, and thank you to all the judges and to you, Jeremy, of course. Before we part, I would like to invite the winner, Leila Saik, to say a few words, and also a word of my own to those who didn't win. I know that it's probably disappointing, but it was really, truly remarkable set of works. So thank you for sharing those works with us. Leila, a few words from you?
Yes, thank you. I'm so excited. I'm honestly sort of speechless. I was not expecting. So I'm very thrilled, and I just appreciate your support of our work. And I really also appreciate the recognition of the value of long-term data sets. We are so lucky to be working in this field site where I actually am right now, where we have, more than 40 years of data that can be used to answer these kinds of questions. So I'm super excited to now have more capability to keep working on it. So thank you.
Thank you, and congratulations again. I guess this is where we part. We just want to again thank all the competitors, the judges, you, Jeremy and Yossi. Again, congratulations to the winners and to all finalists. Many thanks to you all and to the audience for being us today, and see you next year.
Bye, everyone.
Bye, everyone. Thank you very much.
Thank you very much.
Thank you, everyone.
Thanks.
Thank you.
Thank you. Bye-bye.